Okay. We're gonna get started. Hello, everyone. Welcome to the first meeting of the Middle School Youth Book Club. Today we'll be going over. Can everyone see my screen? Can everyone see the slide? Okay. I can't see my presenter. Notes. Okay. So this is what we will be going through today. We'll first fit. I'm so sorry. I'm Anthony. I will be one of the officers teaching you. I will be teaching you today. Angela, say hi, Angela.
Hi. Hi, I'm Angela. I'm your other officer, and I won't be teaching you these slides, but I'll be teaching you guys next week.
So we'll maybe take turns teaching. You'll have one of us every week, and our instructor is Mrs. Wen. Can everyone say hi to Mrs. Wen in chat? Well, I'll assume people, yeah. Okay. Now with that out of the way, I think we can get started with the lecture. Angela, do you have the admitting stuff under? Okay. Are you admitting?
Yeah,
people?
I am.
Okay. So, all right, let's get started with the lecture. We're gonna be covering 3 chapters today. So first chapter, chapter 2, the chemical context of life. Next is chapter 3, a discussion between water and life. And finally, chapter 4, talking about how carbon is very important and diverse in life. So let's get started. Chapter 2, the chemical context of life. So most of you have probably taken some form of science or chem. So I think we know what elements or compounds are. An element is a substance that is made up of one kind of atom. So, for example, oxygen, O2 is an element. Even though there are 2 atoms of oxygen in every molecule, both atoms are the same kind of atom. So it's still considered an element. A compound, on the other hand, is a substance that is made up of 2 or more different kinds of elements combined in a fixed ratio. So sodium, that's probably not the best example. Carbon dioxide, CO2, is an example of a compound. It has the atom carbon, it has the atom oxygen in a fixed 1:2 ratio. Will these slides be shared? Angela, do you know? I think we can share these slides.
This session's also recorded, so all of the slides can be on it.
Oh, did I start recording?
Yeah, you did.
Okay, good. Regardless if we share the slides, you can see the slides through our recording. Moving on, there are certain elements that are more important to life than others. These are called essential elements. These include carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, or as a lot of people like to use as an analogy, chomps. These make up like maybe 97% of all living matter, very important to life. On the other hand, the other like maybe 3%, 5% is made up of trace elements. These elements you need them in very small quantities, often as cofactors, meaning that they assist in enzyme function. You only need a tiny bit of these, but you still need them regardless. And if you don't have trace elements, you can get very sick. And then a little bit about atoms as just atoms. Every atom has an atomic number, which is basically the mass of that atom. Now the atomic number is made up of, is the sum of the number of protons and the number of neutrons. Should you write these down? It's up to you. I think, here's what I think. If you're having trouble understanding the material, I would suggest writing it down. If you are somewhat comfortable with it, then I would just suggest following along with the lecture. But it all depends on whether or not taking notes helps you remember and learn as an individual. And yes, this session is recorded. Okay, so sorry, I meant the atomic mass. The atomic mass is the sum of the protons and neutrons. That's a mass of an atom. Whereas the atomic number, my apologies, is the number of protons in an element. The number of protons in an element distinguishes, it tells you what kind of element that is, because every element has its own specific number of protons. If you see my mouse, I'm circling over this notation. This is what we generally use to denote an atom, or to subscript, to shorthand an atom. So right here you have the symbol for the atom, in this case carbon. On the top left, we have the mass number, in this case 12, and in the bottom left we have the atomic number, in this case 6. Can anyone tell me how many neutrons this carbon atom has? Type it in chat. Type it in chat. All right, good. You guys are getting it. Because the mass number is the number of protons plus the number of electrons, and the atomic number is number of protons, we know that there are 6 protons in this atom, which means that there are 12 minus 6, there are 6 neutrons in this atom. Is my audio good? Are you guys hearing me all right? Okay, cool. Now moving on. So a little bit more about the subatomic levels, subatomic particles. Every atom has electrons. Electrons are little bits of negatively charged particles that kind of, you can think of it as orbiting around the center, which is the nucleus with the protons and neutrons. The electrons within the electrons that are closer to the nucleus have lower energy. The electrons that are farther from the nucleus have higher energy. Now, the way that atoms interact with each other is mostly through bonds. So they will use their electrons, their outermost electrons, to form bonds with each other. These outermost electrons are called valence electrons. That's probably something that you should note down if you don't already know. Yeah. So in this case, these 2 valence electrons are going to be shared between these 2 atoms and form a covalent bond. We'll talk about what a covalent bond is later. Formation of molecules. A molecule is a group of 2 or more atoms with electron interactions. And then there are 2 types. Speaking of what we just said, there are 2 types of bonds. One is an ionic bond, one is a covalent bond. So can anyone tell me what is an ionic bond? Okay, that's a generalized, yeah. Generally, ionic bonds are between a metal and a non-metal. The specifics of how it actually works is you have one atom that really wants to get an extra electron, and you have another atom that really wants to give another electron. So the atom that really wants to give an electron transfers an electron to the atom that really wants to get an electron. As a result, these 2 atoms now have opposite charges. And because they have opposite charges, opposite charges attract, so they'll come together. So it's not actually the transferring of electrons that creates a bond. It's the resulting charges that allow the atoms to stick together. In regards to Aspen's question, is this chemistry or biology? There's a lot of chemistry involved in biology, especially when you get to the higher levels. You'll learn that these two fields are very interconnected. So I guess you could say strictly that this is chemistry, but chemistry is also part of biology. Biology is part of chemistry. It's hard to draw the line. Okay, the other type of bond is arguably more important, in my opinion, for light. It's called a covalent bond. So this is a bond between two nonmetals. So carbon-carbon is a covalent bond, but sodium chloride is an ionic bond. Instead of transferring electrons in a covalent bond, the two atoms share electrons. So they both like contribute 1 or 2 electrons and form a shared bond. However, in this case, because the electrons are still like shared between the two atoms, they're not taken strictly away from one atom, right? Therefore, a property called polarity can occur. Who knows what? Type yes in chat if you know what electronegativity is. Okay, a few people. A lot of people. Okay, so I'll just explain it. Electronegativity in a covalent bond is how much the one atom wants to take the bonded electrons. So let's say you have oxygen, which is very electronegative, and hydrogen, which is very non-electronegative. If hydrogen and oxygen were to form a covalent bond, because oxygen has a higher electronegativity, oxygen will kind of pull the electrons towards itself. If in that case, does oxygen get a partially positive or negative charge? Yeah. Yeah, everyone's correct. Oxygen, because it basically gains more electrons because it's pulling electrons towards itself, right? It becomes more negative, and as a result, it has a partial negative charge. The hydrogen gains a partial positive charge. So this difference in charges within a molecule is called polarity. And this concept, polarity, is very important with hydrogen bonding, which we'll discuss in just in 1 second. And a little bit about metabolism, chemical reactions. There are, you can generalize all of metabolism into 2 categories. There's anabolism, which means you're taking simple molecules and you build them into larger molecules. So like, let's say you have a bunch of amino acids floating around in cytoplasm. If you take those amino acids and put them together into a protein, that's catabolism, because you're taking smaller units and building them into a big one, right? Catabolism is the opposite. That's basically, imagine you have a glucose molecule, which is pretty big, has a lot of atoms, and you take that glucose, you break it down into carbon dioxide and water. That's catabolism. You're taking a complex molecule and you're pulling it apart into small, simple molecules. In general, anabolism will result in will need an energy input. So it will result in a net negative energy. Does that make sense? Like, to make anabolism results in net negative energy. Okay. If you don't understand, send me a DM or something. Catabolism, on the other hand, like the glucose example, if you break down glucose into water and carbon dioxide, you get a release of energy. So catabolism is associated with a Angela. Catabolism is associated with a release of energy, right? All right. Waiting room. And that's it for chapter 2. Any questions? Shoot me a DM if you. Ivan? Okay. Okay. It doesn't seem like there's any questions. Now we're gonna move on to chapter 3. This chapter is about water and how it's important to life. Does anyone have all of these? Every single one of our slides is gonna be based on the book. So if there's something you don't understand, you should go and read the textbook. If you should, I would even say that maybe like skim the textbook ahead of time so that you understand what we're talking about in the lecture. Today, most of the stuff is pretty rudimentary, so it's not that necessary. But yeah, everything is from the textbook. It actually is Ivan. He showed up. Ivan is our club president. Everyone's say hi to Ivan in the chat. Hi, Ivan. Even though he's okay, you gave me this account. I don't know what to say, man. All right. Now let's get started. We're gonna review a topic that we just discussed, polarity. Polarity, well, we just discussed this, right? Does anyone want to try to explain? Actually, raise your hand if you want to try to explain this. Raise your hand. Virtually, of course. Okay, nobody. Okay, polarity is, if you can see in this diagram, this indicates a partial negative charge, right? And then this, go away, this indicates a partial positive charge. The differences in charge within a molecule is its polarity. So in this case, the difference between this partial negative and partial positive, it is pretty high. So this molecule has a high polarity, right? Like we discussed before, oxygen has a very high electronegativity, right? So that means it will pull, let's say there are, I can annotate, I forgot. Let's say there are 1 electron here, 2 electrons here. Let's just say, simplify this into this. Oxygen having more protons, having a pretty small atom size will pull these electrons towards it. And as a result, oxygen will gain a partially negative charge. And because hydrogen has its electrons taken away, it gains a partial positive charge. That's called polarity. I'm explaining this again because nobody wanted to explain, so I feel like most people didn't get it. If you don't understand, we have plenty of time. Shoot me a DM. I can go over it again. All right. I'm going to move on. Where do I go back? Okay. This polarity allows for hydrogen bonds to form. So a hydrogen bond is different from a covalent or an ionic bond. In a way, because a covalent or ionic bond is the attraction between 2 individual atoms, but a hydrogen bond is attraction between 2 molecules, right? So in this case, see, the partially positive hydrogen is attracted to the partially negative oxygen of a different water molecule because opposite charges attract. So what this ends up is all of the water molecules are bound to each other by this hydrogen to oxygen connection. There's a lot of these intermolecular forces that pulls water very close together. Okay. And hydrogen bonding is why water is so important for life. That's one of the only reasons that water is this important. It gives water a lot of properties. excuse me. The first one is called cohesion. That's what we just demonstrated. Cohesion is a tendency of water molecules to stick together. In this case, because of the charges, the opposite charges, the water molecules are attracted to each other and they will stick together. So, for example, in a tree, when the video of the slides will be posted, I think we might make a Schoology group later or something. We'll keep you posted. We'll figure out a way, even if it's through like email or something. For example, when a tree brings water from its roots up to its leaves, it does a process called transpiration, right? So what that is it draws a string of water from all the way up to the leaves. And the reason that I can do this is because the water molecules are all attached to each other by hydrogen bonds. So when you pull on the very top water molecule, all the water molecules below it are pulled up by hydrogen bonds. So imagine if you had a small piece of string connected to like a pen. If you have a small piece of string connected to the pen, if you pull on the string, the pen will go up. In this case, the hydrogen bonds is that piece of string, right? The next property is called adhesion. Adhesion is okay. So who can explain how hydrogen bonding occurs? Actually, raise your hand. Raise your hand. It's probably going to take too long to type. Okay, guys. I'm gonna cold call on someone if someone doesn't raise their hand. Who should I call? Ishika, do you hold on? Ishika, can you explain how? Yeah, go ahead. Can you explain how hydrogen bonding occurs, or what process drives it? Okay. It's all right. Hao Tian, is that, yeah, you can.
Sorry, I'm Marco.
Oh, Marco. Oh, I didn't even, okay. You want to explain?
So basically, wait, can you repeat the question again?
How do hydrogen bonds form? Why do they form, basically?
So basically, hydrogen bonds, because like the hydrogen side, like water is a polar molecule, so like the hydrogen side is like positive, the oxygen side is negative. So basically, like positive attracts negative. So yeah, they bond.
Yeah. So thank you, Marco, and thank you, Rocky, and I'm sorry, I'm gonna mispronounce this. Anirudh, for volunteering to raise your hand. I really value participation. Thank you, guys. So the property of hydrogen bonding occurs because water has positive and negative charges. And as a result, water can not only hydrogen bond to other water molecules, it can also hydrogen bond, not hydrogen bond, but it can also attract to other charged molecules in other parts of cells. So for example, if you think of it, one example is like this tree. In this tree, as water is being yeah, go ahead, Austin. Do you have a question? Oh, I'm sorry, you're muted.
I want to get this like clear.
Mhm.
Does a hydrogen bond like kind of like not break so easily, or is it like a bond?
Yes. Well, that's a good question. We'll get to that in a bit, but yes, hydrogen bonds are very strong in terms of intermolecular forces. that's another reason why the properties of water occurs. Yeah. Okay, thank you for the question. And adhesion is a tendency of water. So like, let's say there's a positive, I'll answer that in a bit. Let me finish explaining adhesion. So let's say that there's a positive charge in the cell wall right here. Oh, my bad. Let's say there's a positive charge in the cell wall right here. That will attract the negative oxygen onto the cell wall, right? That's adhesion, the ability of water to bind to other substances because it has that difference in charge. Anirudh asks, isn't oxygen only a bit negative? Why is it so strong? So yes, if you compare oxygen, the partial charge of oxygen to a metal, for example, like potassium. Yeah, sure. I'm really sorry. I don't know how to pronounce your name, but you didn't really miss much, and everything will be recorded, so you can watch the recording. Although oxygen is not as negative as like a potassium atom, for example, you're comparing intramolecular forces to intermolecular forces. So like a potassium atom is just a single atom, right? If that atom bonds to a chloride, that is a very strong interaction, but only because they're very small and they're very charged. They're very polar. I think we are taking a 10. In terms of intermolecular forces, so the attractions between two different molecules, hydrogen bonds are the strongest force there is. Does that make sense? Yeah. Okay, cool. No problem. Ivan, we can get to that in a bit. And finally, because water is held very tightly together by hydrogen bonds, that means that water, like I'll explain this with an example. If you've ever been to a pool, I hope you guys have. It's and okay, bye Ivan. If you ever go to a pool and you slap the surface of the water, it doesn't immediately like give away. It hurts. It's like it feels like you're hitting a solid surface, right? That's because the hydrogen bonds in water is holding the surface of the water, is holding the surface together. And so when you slap down, the hydrogen bonds resist you pushing the water molecules away. It holds the water, it holds the top of the water together. That's why water has high surface tension. Okay. Angela, do you know what how the ID thing works? Do you want to explain? Oh. Okay. Is it just the give me 1 second. I'm gonna stop sharing. I'm gonna pull up the ID sheet. Basically, from next week on, everyone here is going to get an ID number, like a small sub 40 number. And that number will indicate who you are. It'll help us with attendance. We want you to put it in front of your name, like this. So the X is your number, and then you add your name. I will pull up the list of all of the IDs. Give me just 1 second. Okay, I think. Okay, cool. Angela will distribute your meeting IDs. Sorry for the interruption. Let's get back to the lecture. Can everyone see my screen? Oh. Okay, cool. All right, let's get back to it. Oh, I think that's all right. Don't worry about it, Reba. Angela sent that to me. Okay. Water has three other properties that makes it very important. The first is high specific heat. What is specific heat exactly? So the definition of specific heat is the energy required to heat 1 gram of a substance by 1 degree Celsius. In a more general definition, it's how easily you can heat a substance. How much energy does it take to make something hot, right? Water, it takes a lot of energy to make water hot. Can anyone tell me why? Just guess. Just say something, anything. What? A number? Yeah. Okay. Go ahead, Rocky. I'm sorry, I can't hear you.
Hello.
Hi, what's up? I can hear you.
So the reason that liquid water takes many heat to heat up is because of like the structure of hydrogen bonding between the water molecules. Like
Yeah.
because the bond is like slightly stronger than other molecules, it takes a slight more energy to break apart.
Yeah, that's a great explanation. If you ever get a question anywhere about does water have this property, 9 times out of 10 it's hydrogen bonds. If you're gonna remember one thing, oh no, you're all right. If you're gonna remember one thing, water has hydrogen bonds, and that's what makes it special. Okay. Now why is this important? Around large bodies of water, like near the ocean, for example, it's very chilly. That's because the ocean is absorbing a lot of the heat nearby. It's absorbing a lot of heat while only getting hotter by a little bit. And as a result, it's a good regulator of temperature. Right. And another, I'm sorry. Because water has high specific heat, that also translates to a high heat of vaporization. That means that when water evaporates, when water evaporates, it takes a lot of energy to get it to evaporate. So this helps when you sweat. And why do we sweat? Anyone know? Raise your hand. I'll give it like 5 seconds. Why do we sweat? Rocky. Actually, sorry. Let's get someone else. Severe, why do we sweat?
Cause our body is trying to like keep the same temperature, so
To
it's
cool
like
down. Yes.
yeah, cool
exactly.
down.
Good job. Nice. So we sweat to cool down. The reason that sweat is so good at cooling us down is because it's mostly water. And as water evaporates, because it has a high heat of vaporization, it takes a lot of that energy from our skin and it goes into the air. So all of that heat energy is leaving you and making you more cool. That's why water is so effective at cooling things. Yeah. Okay, Rocky. Okay. Yeah. So another property of water is that it expands as it freezes. The reason for this is generally when Severe, do you have a question, or is your hand just still up? Okay, we're good. The reason generally when things freeze, they stop moving and they pack closer together. Like if you have a membrane of phospholipids and you freeze it down really low, all the lipids, they stop moving and they cluster really close together. And as a result, the membrane becomes more dense. But in the case of water, water actually expands as it freezes. And can anyone tell me why? Hands up, hands up. I want to see hands. Come on. I mean, 5 seconds. All right. Properties of water. Come on, I just went over this. If there's a question about why does water have this property? Okay. Yeah, go ahead, Rocky. I can't. Sorry.
Sorry, it's a microphone had a problem. Because of the like the crystal lattice that makes up the water, when it like
Hydrogen bonds.
Yeah.
That's what he's trying to say.
yeah,
Yeah, it's always hydrogen bonds. Always hydrogen bonds. That should come to your mind immediately. Okay. I actually have a diagram of this right here. You see how all of these water molecules, when they freeze, their hydrogen bonds are keeping them at, think of it as keeping them at arm's length. It's keeping them further apart, right? Yeah. Okay, and the last thing is that water can dissolve polar substances. What does dissolving something even mean? So let's look at this diagram, right? Let's say that you drop a cube of salt into just pure water, What will happen is, do you see, this is what your salt would look like originally. Do you see my mouse? What's up, Marco? Oh, okay.
Sorry, nothing.
Yeah, you're fine. This is what your salt would look like originally. And this is what happens when salt dissolves. Basically, what happens is, let's take this sodium molecule, for example, attached to this lattice of salts. What the negative part of a bunch of water molecules will come here, not attach, but pull on this sodium and pull it off of this actually, I can annotate this. So a bunch of positive water molecules will come and attack this sodium, and it'll pull it off, and it'll come over here, right? It'll go this way, and it'll get surrounded by a shell of water. This is called a hydration shell. That's probably something you need to know. I'll type it out here. Hydration shell. This prevents the sodium and chloride from coming back together and reforming the salt. Water will pull off individual ions one by one until everything is surrounded by a hydration shell. So, like, for example, the positive ends of a water molecule will attack this chloride and pull it off. It'll pull it off from the main salt, and then it'll surround it with the positive ends. See, all the ends that are pointed towards the chlorine, the negatively charged chlorine, are the positively charged hydrogen molecule. Okay, clear this. All right, we already saw the thing on the right. So, can anyone raise your hand? I'll give you 10 seconds. What is this an example of? One of the properties that we just mentioned. One of the properties. Aston, go ahead.
Hydrogen bonds.
That's a good. There are hydrogen bonds here. That's an acceptable answer. That's it's not the one that I was looking for, but it's still good. And next time, Anirudh, can you unmute and tell me how to pronounce your name?
You can just call me Ani.
Ani, cool. Next time, Ani got the right answer. This is cohesion. It's the sticking of water molecules to itself. Water sticks to itself from the positive end to the negative end. Next time, just like unmute, tell me. Unless I specifically tell you to send me a private message, I prefer if you raise your hand and unmute. Yeah. All right. What is this an example of? This is a water molecule attached to let's say a cellulose fiber. What is it? Yeah. Anyone else? Anyone else? I want to have a bit more participation here. Okay. Okay, Aston, go ahead. Go ahead.
Oh, wait, which one? I forgot.
This one. Oh, sorry. This one right here. Bottom left. So water attached to something.
Adhesion?
Adhesion. Correct. It's a water molecule attached to a different thing. Sorry, I couldn't find a better diagram. Okay. And now let's talk about the properties of water. A substance that dissolves within water is called hydrophilic. So something that likes water, something that can hydrogen bond with water or has partial charges or full charges, like a sodium molecule, a Na+, it has a full +1 charge. That is hydrophilic, right? That is hydrophilic. Whereas like a lipid or like a fatty acid, which is largely non-polar, it has no like charges on it, will be hydrophobic. It will not dissolve in water. And I can talk about it. You have a question, Aston? And then solute concentration is this is just a term you need to know and a good introduction for what's on the next slide. It denotes moles of solute per liter of solvent. What's a mole? What's a mole as a unit of measurement? What's a mol? For this, yeah. Super got it. Yeah. Nice. Okay.
I feel like we're learning chemistry and biology at the same time.
A lot of the intro things, like these two chapters, will be chemistry. I think it's just the first few. Like next week, we'll probably dive into, I think the cell, specifically, like organelles and stuff. Oh no, you're good. You're good, Ria.
Wait, hold up.
Yeah, what's up?
The
Ohio.
question.
Ohio.
Yeah. Go ahead, Marco.
So, isn't this like? I'm sorry, because I was here like last year for like the middle school bio board, but I just realized that you're talking about the cell. Isn't this like what I wrote like earlier was like macromolecules and stuff? Is there a reason why this is simpler?
What do you mean? Like simpler
Like
than the
this
cell?
simpler than last year.
Simpler than last year. I'm not exactly sure. I didn't teach middle school last year, so I'm not exactly familiar with the curriculum. But we will cover the same materials. That's I think that's for sure. And I think we will never be able to cover everything in the book, like in detail. So if you really want to learn more, I think reading the book is the best way to learn.
Okay.
Yeah, that's just what I do. So, yeah. And now let's talk about acids and bases. So acids donate protons and bases accept protons. Do we all understand how that works? So the acidity of a solution is a measurement for how for how much acid there is in a solution. It's a measurement for how much acid is in the solution. Specifically, it tells you the concentration of hydrogen ions. This is biologically important because in general, things with single charges are dangerous to have just floating randomly around in a cell. Especially because pH affects protein function a lot. But pH tells us the concentration of hydrogen, right? But I'm sure most of you are familiar with pH as just a single number. So how does that work? I think, okay, go ahead.
Wait, no.
It's all right, it's all right.
Oh, I thought you said my name, so I was like,
No. You're good. So, let me illustrate this with an example, okay? So let's say your hydrogen concentration. Oh, by the way, if there are brackets around something, if there's brackets around this, that indicates the solute concentration of X, okay? So in this case, if your solute concentration of hydrogen is 10 to the -3 moles per liter, that means that your pH is 3, okay? And if you have a pH of 5, that means your hydrogen concentration is 10 to the -5 moles per liter. If your pH is 9, that means your hydrogen concentration is 10 to the -9 moles per liter. Does that start to make sense? So it's whatever the concentration of hydrogen within a aqueous solution is always going to be 10 to the power of negative something, and that something is your pH. So let's say you have a solution of 10 to the -4 moles per liter of hydrogen. What is the pH of that solution? DM to me. Oh, it's all right. Don't worry about it if you didn't DM. Yeah. Hey, I'm consistently getting the responses from a few people. Does that mean that the rest of you don't understand what I'm saying? Or does that mean that you guys just aren't participating? I'm not going to cold call you. I just want to make sure that everyone's understanding what I'm saying, right? Like we have plenty of time. We can go over, or yeah, okay. If you have a question about something, even if you even if you don't have a question, you just don't really understand something. You can tell me to go back and I'll try to explain it again, and hopefully it'll make more sense. Yeah, it's all right if you're writing notes. I'm not blaming you. I just want to make sure that you guys understand. Okay, let's speak. I haven't been talking about how much we. Okay. Yeah. This is just a fun fact. I'm not sure if you guys need to know this from middle school, but if you multiply the concentration of hydrogen and the concentration of hydroxides together, it'll always equal 10 to the power of -14. So look at my screen. I pause for a second. Look at my screen. You see how in all of these cases 10 to the power of -3 multiplied by 10 to the power of -11. If you multiply all of these values, they all equal 10 to the power of -14. And that's just a general thing. So if I told you that there's a solution with a pH of 6, what's the hydroxide concentration of that solution? You guys, this is going to require a bit of reverse engineering. Yep. Ani got it? All right. So a pH of, yeah, good job, Severe. A pH of 6 means that you have a hydrogen concentration of 10 to the power of -6, right? A pH of 6 means that you have a hydrogen concentration. Oh, I sent that to sorry. Will bio Olympiad include things like human anatomy and ecology? Angela, do you know? I'm pretty sure it will. Those are pretty basic parts of bio. So if you have a pH of 6, that means your hydrogen concentration is 10 to the power of -6. That means that your hydroxide concentration has to equal 10 to the power of -8, because yeah, a lot of people are getting it, because if you multiply those two together, you get 10 to the power of -14. Are you raise your hand if you don't understand how this works. I can go over it again if people don't understand. I don't really, if you ask for the mechanism behind this, I'm not 100% sure. I think this is just a general thing, like a fun fact you need to keep in mind. Why exactly this happens, I'm sorry, I can't tell you. I think that goes more into like harder chem. I'm a bio person. I don't know chem. Yeah, and calculate what? Calculate what? The concentration of hydroxide. All right. Okay, so I'll go over it again really quickly. So let's say you have a pH of 6, right? A pH of 6 means that you have a hydroxide concentration of 10 to the power of -6. Because that's the definition of pH. This 10 to the power of whatever is your pH, right? You know that concentration of hydroxide times concentration of hydronium must equal 10 to the power of -14. So 10 to the power of -14 equals 10 to the power of -6 times 10 to the power of -x. And this is just and then it just turns into algebra. You divide by 10 to the power of -6, and you get that x equals 8. So the concentration of hydroxide equals 10 to the power of -8. Does that make sense? Okay, no problem. And finally, there are things like buffers that prevent excessive acid-base chain fluctuation. Like, for example, the reason this is important is like if your blood pH increases by like 0.5 or decreases by 0.5, you die. That's it. So it's very important to have buffers in your blood to make sure that your blood pH doesn't go too high or too low so that you stay alive. To reiterate, buffers are basically, they're just things that prevent huge pH changes. So if there's a lot of hydrogen in the solution, it'll absorb hydrogen. If there's not much hydrogen in the solution, it'll give up hydrogen. It's fine. Nah, don't worry about it. Don't worry about it. We have plenty of time. Okay. We don't have many slides in chapter 4, but there are certain important carbon formations that you generally need to know. Anything that's made up of carbon and hydrogen is a hydrocarbon. So like a lot of things in biology are built off of a carbon skeleton, meaning the base of everything is carbon, and then you attach things to carbon to give it special properties, right? Hydrocarbons are generally hydrophobic, so they don't dissolve in water. And functional groups are the things I was talking about. You add these to carbon skeletons to give it special properties. So if you have a short carbon skeleton and you tack on a polar functional group, so that will make the molecule more polar. If you tack on a non-polar functional group, it'll make the molecule more non-polar. So the functional groups give things special properties, is what I'm getting at. here's a great example of why the book is important. That these are these are some of the functional groups that you need to know. These are all that are listed in the book. Polar increase, and why don't they just call it polar increase and polar? Could you? I'm not sure I understand your question. Could you like rephrase it? All right, back to this. Functional groups are there. You should know their names and their polarities. There's a huge chart of all of these functional groups in Campbell's, and there's a huge chart of all of these functional groups in Campbell's. And if you want to know more about them, you should look at the chart. Ani asks, why are some bonds empty? That's a great question. Good eyes. These empty points indicate that this functional group is attached to something else here. So like, this is a point where hydroxyl attaches to a carbon skeleton. Like this, it could be anything. So it just leaves it as empty. Riva, do you wanna say your question out loud? Is that what you're trying to say? You can go ahead. Why don't they just call it polar increase and polar decrease?
Are we ending this meeting at 8?
We might go like a few minutes over. Actually, no, that the next slide is our last slide. So we might go like 2 minutes over time, but if you need to go, you can go at 8. We just have one more slide though. So Riva, you can stay if you want, you can stay after, and then you can ask your question again. Okay. I'm just gonna get through this last slide so that people can go. All right. Finally, about macromolecules, about any molecule in general, there are things called isomers. Isomers are two different molecules. They're two different molecules that are made up of the same atoms, but they have different like physical geometrical structures. So the first type is a structural isomer. If you look at this, pentane versus 2-methylbutane. Pentane, these two molecules have the same kinds of atoms and the same number of atoms, but they're different because they have a different molecular shape. This one has a bend, it has a fork. And that's a structural isomer. Next is a cis-trans isomer. So a cis-trans isomer arises from the fact around a double bond, you can't rotate the atoms. Around a single bond, you can like spin things as you like, but around a double bond, everything is like locked solid. So for example, if the two x's are on one side, then this x can't move over back, can't flip to this side. And so that gives these specific properties. For example, so if the two x's are on one side, that is a cis isomer. If the two x's are on opposite side, that's a trans isomer. And finally, there are enantiomers, which they have the same molecular structure, they have the same atoms, they have the same almost everything, except they're like mirrored of each other. So like if you take your two hands, right? Both hands have a palm, they have 4 fingers, a thumb, but they're not the exact same. Why? Because if you put them together like this, you have a right hand and a left hand, right? I'm sorry, Aditi, I saw your question. I'll get to it right after I finish it. You have a right hand and a left hand. They're made up of the same things, but they're not the same because they're two different hands. One has the thumbs are on different sides, but if you mirror them, if you put them on a mirror plane, they match up perfectly. That's basically, think of two enantiomers as your two hands. They look the same, but they're positioned differently. All right. All right. I think those are all the slides that we have for today. Thank you all for coming. You are free to go. I hope you guys learned a lot, and take care.
Thank you. Bye.
No problem.
Yes, thank you so
Bye-bye.
much. See you
Okay.
next
Thank
time.
you. If you have questions, you can stay behind.
All right. See you.
Oh, okay. Bye. Bye, Riva. If you, is Riva still here? No, she is not. Okay. So, Ani asks, so there's nothing different in isomers except for the shape, right? Basically, there's nothing, all of, yeah, they're made of the same kinds of atoms. They're made of the same number of atoms. It's just that the atoms are positioned different. Yep.
What should you study next week? Well, next week, I'll be talking about more hydrocarbons, and then wait, no, that's already talked about today. Okay,
Okay.
I'll be talking about macromolecules, so it's like proteins, carbohydrates, fats, like
What
lipids,
chapters?
etc. And then,
Chapter.
5
I'll
to
pull it up. Okay, so next week we will be covering chapters 5 through 7 of
yeah.
Campbell's. I think
This week, should I study 1 to 4?
I think next week you should study, like, skim through chapters 5 through 7. These they're pretty big chapter, like not big, but like each Campbell's chapter is pretty big. So I would suggest like just skimming through it, looking at key terms, like trying to read as much as you can without interfering too much with your schoolwork, right?
Yeah.
Also,
So
look at
yeah.
Also, should I look at the questions like at the back of the chapter?
that's good review. That's good review. Do you just read through the macromolecule section of the textbook, or do you recommend any other studying technique? For me, all I do is read. That's the only way I study.
It's good.
I'm sure that if you looked on YouTube, there are probably some good videos explaining things. I don't
Oh, can you
really.
channel name?
What's up?
Can you share the channel's name?
Sure. Oh, no. I just meant like in general. I don't have a specific channel in mind.
Oh,
My apologies.
and also, are we going to cover the whole book this year?
The whole book. I think we plan to. Yeah. Yeah, we should. Okay. Okay. Bye, Ani.
Thank you. Bye.
No problem. See ya.